Welcome to the Molecular Moments podcast.
In today's episode, we sat down with our guest, Dr. Laura Sepp-Lorenzino, Chief Scientific Officer at Intellia Therapeutics. Laura is an innovative leader in drug discovery and development of small molecule and oligonucleotide therapeutics. She's also led vaccine development programs, and more recently, she has made her mark in the exciting field of gene therapy and gene editing. She has over 30 years of experience in academic and industrial settings. I first met Laura several years ago when she was the keynote speaker at the Land O'Lakes Bioanalytical meeting, and I've been a fan ever since. I hope you'll enjoy our conversation as much as I did. We're talking scientists as scientists do. So without further ado, here's the 6th episode of Molecular Moments. Welcome to the podcast, Laura. I'm delighted to have you join me today. Can we start with you giving us a few highlights from your career?
Chad, thank you so much for having me today. I've been listening to your Molecular Moments podcast, and, it's really, really very engaging and informative. So, I hope that today's episode will meet that mark. So I'm a biochemist by training. I was born, went to school in Argentina. Then I came to the US to do my Ph.D. and postgraduate work. And then I've been working in industry since 1999, so long time in industry. First at Merck, then I went to Alnylam, Vertex. Now I'm the Chief Scientific Officer at Intellia Therapeutics. Really having each of these experiences, build, my professional experience, but, it's all of them were different, and it gave me the opportunity to learn different things from, the different companies, and, I'm still learning. This is what makes it fun.
Laura, I'm really excited to explore that with you. And one of the topics that I'm curious about is how you made this transition coming from Argentina to the United States for graduate school, and what kept you here. And tell me about what brings a girl then, I guess pretty young, here to the U.S.
So I studied biochemistry, pharmacy and biochemistry at the University of Buenos Aires. And although my mom is a pharmacist and her wish was that I was going to take over the family business, I was always drawn towards research. And I started doing my Ph.D. in Argentina. And my boyfriend at that time was going to start his Ph.D. in the US. So his sister, who is smart and very practical person, suggested that maybe we should do the PhD in the same geographical location. And, and I quit the program in Argentina and I joined him in coming to the US. So we were both newlyweds. I think we were 2 weeks after our marriage when we both started graduate school. So it was, quite an awesome experience, right? Starting, our life in a different country, different language, and both of us pursuing our dreams, At that time, he was a chemist. I was doing biochemistry. It was really great to have each other to help, support and push us forward, right? I can tell you that, and anybody who has immigrated can share this, that, as you go to a different country, different language, different culture, you encounter, a number of challenges. And, it's good to have someone as your partner to help you figure out how to navigate those new challenges together.
Well, you may not have ended up as a pharmacist, but I have to believe your mother would be proud of you for what you've achieved in drug development. So your mother was a pharmacist, and then you wanted to do research and move forward in that direction. It's a little bit different direction. But are there any other personal connections or anything else that really drove you into science, or maybe why research versus taking up the family business?
Yeah, and I think that mom is responsible for that as well. So I grew up going to her pharmacy and, she had books and magazines. So, even as a young child, I would flip through all of that and make believe, that I was contributing to those journals, right? But then one day she took me to a seminar, and you'll appreciate that. They were showing a biodistribution of a drug, right? And they were doing this whole body autoradiography on, the distribution and disposition of a drug. And seeing how that, labeled drug, was taken in the animal, distributed, how it made it to the target organ, right? And I was 9, I was really young, but, the guy who explained it, somehow made it really understandable. And that's when it clicked. It was like, this is so awesome. Nothing compares to this. I really got lucky because I had people who helped me, with opportunities and mentoring, and this has been invaluable to me, and it's so important for us now to pay it forward, right? To do the same for the younger generations.
Without a doubt. So I want to jump right into some of the science, and I'm going to go right into what you're working on now in the space of gene therapy and gene editing. And maybe you can give a 3 to 5 minute primer on what's gene therapy versus gene editing, and kind of how those play in with the modern science. Because I'm thinking when you were in that pharmacy, in your mom's pharmacy, you probably weren't thinking about, wow, if there was a drug that could change your genes, So tell us about that.
Yeah, not at that time. Yeah, so gene therapy and gene editing are a new class of therapeutic modalities. And here we're aiming to treat diseases at the genetic level. That is, the DNA level, which is the source of instructions that tells a cell how to behave in the context of the organism. What happens is that when those instructions are missing, or they're wrong or incomplete, that's what leads to disease. With gene therapy and gene editing, we are aiming to reset those instructions. And, there are a number of analogies of, using a word editor or writing computer code that, I think that they are really good illustrations to make the point. So with gene therapy, you're not changing the master code, but what you're doing is you're adding, a small program that, runs in parallel and sends new or overriding commands to address, this disease-causing event. What we're doing with gene editing, and this is what we're doing at Intellia Therapeutics and other companies in the space, is that we're actually going to that master code and we're changing the information in the appropriate line. So we're either deleting an entire command line, or you could add a new one, or in some cases, if there is a typo, right, we could just go and precisely, specifically make that, small change that at the end is, the driver of disease. Now moving on to the language of, genetics. So the deletion, it's called a knockout. So you eliminate a gene that's not functioning or hyperactive. You could do an insertion or a knock-in where you're adding a gene that has been missing. And in some cases, we can do correction. And this is, particularly when there are very defined mutations that, we can use some of the new tools to go and fix that. So, with these approaches, in particular with gene editing, because we're changing this master code, the goal that we're aiming is that this is going to be a permanent change in that cell, and we're looking to have potentially one and done curative treatments. And we can discuss a bit more about, that.
Yeah. And I'm going to jump to something that scares a lot of people about this, right? Earlier, when we were talking before the podcast, you heard a reference to the superheroes. I was talking with a previous guest, with Jim McNally, and said, can we create superheroes with our gene therapy? But in reality, what really does make a lot of people nervous about gene therapy is either side effects or misusing the technology. And so could you talk about the potential side effects and what we're doing, and why this really is a safe tool for us to use to cure people? Like you said, it's a one and done cure. So
Yeah, so I think that there are two layers in your questions. One, and one is the ethical aspects of this. Are we going to be creating superheroes with, special powers? This is not where we're going, as companies, as a field, as a scientific community. Although, we heard in 2018, right, about these scientists in China editing embryos, right? There is a moratorium on doing that, and, we all ascribe to that, right? I think it's right now we're doing somatic editing, not germline editing, and we're addressing disease, nothing beyond that. So then the second part of the question is, how safe is this, right? Because this is, gene, so we're modifying the genes, right? So there are a number of molecular tools that allow us to precisely design the effectors, and I'm talking more about, CRISPR, how the CRISPR machinery is going to find the exact region in the genome where we want to introduce the edit and nowhere else, right? So you could use a number of tools in silico and in the lab to figure that out. But also we have a number of robust, very well characterized and accepted by regulatory agencies on how we are experimentally evaluating the on-target versus the potential off-target effects. And the goal, as we are developing human therapeutics, is to have these therapeutics present no off-targets at doses that are going to be used in the humans, in the patients. And to that you add, safety margin, right? So it's our goal is to find the safest, more efficacious candidates as possible. As I was saying about regulatory agencies, right? So, the regulatory agencies in the US and Europe and, other countries, they have been learning and partnering with the scientific community to understand how to do this and what to require from companies that are developing therapeutics to ensure that the highest standards are being met. So there is, data now coming from, for example, sickle cell disease and beta thalassemia using CRISPR-edited hematopoietic stem cells that have really transformed the lives of, the patients that have received them, right? And there is no ill effects. We at Intellia, we have a, large number of preclinical studies we have done. And now we're in the clinic with our first systemically administered CRISPR drug. So, as more companies and ourselves continue to advance these therapies to the clinic, I believe that, the data is robust and we're going to appease some of these potential concerns that, people may have.
Yeah, and I think you touched on exactly what I wanted to mention next, is that when you think about the diseases that we're really and truly curing, a few years ago, these seemed like diseases often without a cure. And typically when I think of gene therapies and gene editing, we see a lot of treatments in the oncology space, of course, rare diseases, and also in eye diseases. So maybe you could touch, again, there's it's a multi-layer answer there, but maybe you could just explain a little bit why those are the spaces where we see gene therapies being so effective for people.
So when we're thinking about, how to deploy these new technologies, right? So one of the considerations is, first, what can the technology do to fix a particular biological problem? And are there other therapies available today that are, providing benefit to those patients? If the answer to that is no, right, there is this unmet medical need, right? So, and how can you help those patients? For new technologies, you look at opportunities where there is high unmet medical need and where the potential risk of taking a new therapeutic modality are outweighed by that, potential benefit that those patients are going to receive. And I'll draw from, one example from gene therapy, right? For example, for spinal muscular atrophy, this is a genetic defect in which for type A, the patients will die by, a year of age or, very young through the work done by Avexis and now by Novartis. These kids are living. They're riding bicycles. It's really game changing. And this is, for me, a good example of, huge unmet medical need and how a novel therapy was really able to transform the lives of these patients.
You could see it as a next step to a Star Trek type world where it seems that nobody is sick, right? I mean, that's I'm a big Star Trek fan, so
I am too.
Oh, good. Well, we're gonna hit that later then. So you touched on something that I'm actually curious about, because my background is not extremely strong in the gene therapy space. Is preclinical. So traditionally, a preclinical program supporting a pharmaceutical development, you're giving the same drug to a couple of different species, large and small, and you look for toxic effects. And you know that for the most part, the animals are going to metabolize it in a similar way to humans. And we've got it worked out how to translate those effects, and it's pretty straightforward. But in a gene therapy program, what does a gene therapy preclinical program look like?
So for any of the, and this is a big difference with small molecules, right? So, in small differences and similarities, right? Because in a small molecule for your tox species, you want to make sure that, your small molecules will have activity against, the rodent gene or the rodent protein, right? So for gene therapy and for gene editing, in particular, more of what we're doing. So, the standard preclinical packages, right? So you do need to look at toxicity, understand the sources of toxicity that come from the drug format, all the elements that make up the drug. And we can talk about delivery in a few moments. The only thing that's different, for example, for gene editing, as we were just discussing, is this in-depth characterization of the on-target and potential off-targets and showing that, there is nothing that could lead to potential toxicity. Because the sequences, the DNA sequence is often not conserved between species. If they're conserved, no problem, because you could use your clinical candidate in non-human primates or rodents, and that will be perfect. In some other cases, you may want, depending on the regulatory agency's advice, right? You may want to have a surrogate to at least to understand exacerbated pharmacology, right? And that's really helpful. But there is nothing very different from what you would do from other therapeutic modalities. The other aspect of this is that for platform technologies in which the components are basically the same from program to program, there is a lot of information that you can draw from the past. And let me explain this a bit better. So for delivering these nucleic acid therapies, whether it's gene therapy or gene editing, in the majority of the cases, you require what's called a delivery vehicle. This is either a non-viral formulation, like a lipid nanoparticle or a polymer, or a viral formulation. And the thing that changes is what goes inside. Right? So once that you understand the biodistribution, the ADME properties of that delivery vehicle, right, that's going to be the same independent of the cargo that they have. And that characterization, translates from program to program. And that's one of the really nice aspects of these modular platforms, right? Because you understand what to expect. And then what's different is obviously the pharmacology of the gene or edit you're trying to elicit.
But it sounds like it's a little bit of plug and play in a sense. That'd be a real simplification of it. But
yes, yeah. And that translates also on the CMC, right? On the technical operations and all the analytical tools that you need to develop to characterize all the components, the drug substance and the drug product. So there is a lot of synergies that are gained from some of these modular platform technologies.
Yeah, without a doubt. So you touched a little bit on the delivery, and it sounded like you have more to say on that. I think the delivery of these therapies is one of the most exciting aspects of how they function, and it's something that we get the opportunity, talking to different companies within BioAgilytix, how they're going about it and why they're going about it using different adenoviruses and things like that for delivery. So maybe you want to touch on that a little more?
Sure. Yeah. So, and I'll start with how we're approaching it at Intellia. So, using gene editing, first, there are two major classes of uses. One is to take cells, putting them on a dish and introducing the edits, that rewiring of the instructions that you want to have in your cell product. And then you could introduce that into the patient. And, for that, there is a number of established and more novel delivery technologies. But there is the cell that is the therapy, right? And the applications for that is for immuno-oncology, for autoimmune diseases, for hematopoietic stem cell diseases like sickle cell disease, and regenerative medicine. The other big category is the use of CRISPR-Cas9 or gene therapy, genes replacement in vivo, where now you're administering these, in our case, the CRISPR machinery to a patient. And you could do that in several ways. One is that it could be local administration. For example, subretinal injection in the eye, right, for some of these inherited retinal dystrophies. Or you could give it by inhalation to go to the lung. Or in the brain, or intrathecal delivery for local administration to CNS. We can also give it by systemic administration, and that's by an infusion, intravenous infusion, where you're delivering it to the bloodstream, and then you need to make sure that, that delivery vehicle makes it to the organ, the target organ, and then to the cells in that target organ. And this last approach is what we are using at Intellia for our in vivo portfolio. For that, we have chosen to use a non-viral delivery system that's called a lipid nanoparticle, or LMP delivery.
Kind of like a ball of fat, if you really want to.
If you want
to really make it simple, lipids are fat, so it's like you're delivering a gene therapy with a little ball of fat. Yeah.
So this is like eating a big hamburger with a gift, and
only it's healthy.
The gift that comes into those little, globules is, in our case, the CRISPR machinery that will make the edit, and the body recognizes these fat globules as dietary fat, so it will process them the same way. So it's a really great means of delivering nucleic acids to the hepatocytes in the liver. And this is the technology we're exploiting. And there are a number of advantages. The delivery, lipid nanoparticles are not immunogenic, so there is, all patients would qualify to get them. They're safe. It's a clinically validated approach. There is a drug called Onpattro developed by my former company, Alnylam, that's, it's a commercial product because it's a chemical synthesis, it doesn't have the complexities that making viruses do, right? So you could make tons of very homogeneous particles. And, and we can there is a lot of chemistry that we can apply to make them do different things, take different cargoes, potentially go to different organs, and that's what we're working on. Other companies are exploring viral delivery as their main, for their, first products. Most of them have for systemic or local administration use. People use adenoviral associated vectors or AAV. They have different tropisms. So there is a number of serotype. It's a class of viruses with small differences on their capsid. And those differences result in the viruses being able to go to different cell types in the body. So, some go great to liver, others go to muscle, others, work really well in the eye or in CNS. So that is a great advantage of that technology. Some of the disadvantages is, there is pre-existing immunity to the AAV. So there is a percentage of the population that will not be able to receive the therapy because they have neutralizing antibodies. Fortunately, after people get a dose, they will. If they didn't have antibodies before, they will now. So redosing is an issue. And it's effective, but, could be a lot better, right? So, for example, with lipid nanoparticles, we can get to every hepatocyte in the liver. With AAV, that number is much, much lower. And then the complexities of making a biologic, right? So there, the product is the process you follow, and, that's an art.
Yeah, making the DNA is fairly straightforward, but making the complete drug and assembling it into a deliverable product is, well, it's a whole new different science in itself, I guess. I want to talk a little bit about vaccines with you, since that's obviously a hot topic right now. But I have one more question. I want you to think into your crystal ball. Quite often always with these gene therapies, we're repairing a very small piece of your DNA, even, a single point mutation, or we're, repairing a deletion. But there's so many more complex genetic diseases. I'm curious, how far do you think we can take this DNA repair, or how far should we take this to solve more complex chromosomal disorders?
So at Intellia, I'm really excited about our insertion capabilities in the liver. So there we can put entire genes that have been either missing or, are not functioning in the liver. And we're doing that for hemophilia A and B with our partner Regeneron, and then alpha-1 antitrypsin deficiency. And we can get really unprecedented levels of expression in non-human primates. So you could put really large pieces of DNA. As we're building the proof, right, that this technology works, there are lots of advances, new ideas and new technologies on how to introduce different edits or do gene therapy more efficiently. Part of my job as, the CSO is to have a good pulse on what are the tools that we need to have in our toolbox for us to be able to do the right type of genetic change for the right type of disease. So, it's not that, if you have a hammer, that's the only thing you need. You need to have, a complete toolbox. And I think that through that toolbox development, we will be able to make more sophisticated and, and really expand the utility of these technologies, even to the point of going after complex genetic diseases that go beyond, one gene, right? These monogenic diseases. If there are, chromosomal number abnormalities, can you fix that? Today, not yet, but yeah, I would think in the near future, 5 to 10 years, I'm confident we're going to be making progress on that as well.
All the diseases you mentioned that we are able to cure or close to a cure on the curing hemophilia, right, at a base level where it's just gone, right? I mean, imagine that in the many other diseases. So I'm really excited about that. You have a background in vaccines. honestly, vaccines haven't been very exciting for a long time, other than immuno-oncology, right? Immuno-oncology aside, but vaccines haven't been all that exciting for a long, long time, right? And it's programs that a lot of companies have just kind of, they kind of kept them along because it was a good thing to do, and and they probably made some money, but that all changed with the pandemic. So tell me a little bit from what you've seen, how vaccines have changed over the last 15 years or so.
Yeah, and let me qualify my experience here. So when I was at Merck, I had the opportunity to be part of the vaccines research group. So, I learned from a really talented, team. John Shiver, who's now at Sanofi Pasteur, was, my boss and the leader of the group. And Catherine Jansen, who is leading the Pfizer, she was at Merck too. So it was, really great, knowledgeable group of people. So vaccines, people have taken vaccines for granted, to what you were saying, right? For me is, vaccine, without doubt, is the most impactful achievement in medicine, right? So if you're looking at what has really saved lives and eradicated vaccines, that's vaccines, and take it from Jenner's, vaccine for smallpox to, now that was eradicated, to all the, childhood diseases, when I was a little kid, people get measles, and now it's gone, right?
I was actually telling my son just yesterday about my mom sent me out to catch chickenpox when I was maybe 4 or 5 years old. Go get chickenpox now because it's convenient, right?
Exactly.
And he's like, well, nobody gets chickenpox. I said, yeah, that's because we all get vaccinated, Jack.
Exactly. Like, you haven't entered kindergarten, so go get it because we don't want you to miss school.
Yeah, exactly. Right.
How things have changed. So,
yes.
Particularly, I'm very excited about the mRNA lipid nanoparticle vaccines for obvious reasons, right? I mean, this is what I'm working on. The example of how, BioNTech and Moderna, right, have rapidly, identify potent vaccines for COVID-19, the scalability, the speed, the efficacy, and how now that's also being applied as, these variants of the virus are emerging, how quickly they can react, right, and modify, the vaccine, as needed. This is, is a leap. Kudos to my colleagues doing that.
Yeah, neither of my parents were scientists, so when they were fortunate enough to get the first shot of the vaccine, and I think my mom gets her next shot on Saturday. She's a week ahead of my dad because she got the Pfizer. So she's excited about that. But I was telling her when she got the vaccine, she's like, okay, so how long will it take before I have antibodies and how's that work? And I said, well, first get your vaccine and then visualize your ribosomes working hard, right? cranking the RNA and making the protein. So then your immune system can kick in and do their job. And so I was trying to give her a little push, right? Like visualize what your immune system is doing to fight this virus. So I like that you qualified your experience a little bit in your expertise, because I've seen that with a number of the scientists I talk to who I absolutely consider experts. But then as soon as you see vaccine, I say, oh, Laura's definitely an expert, and you are, right? But there's different levels. But I get asked all the time also those questions because they know I'm a scientist. But for Pete's sake, I'm a chemist, right? So we all have become virus and vaccine experts. One of the things that's really exciting with the vaccine development, and you touched on, is how rapidly those were developed. The groundwork was there, but the disease didn't exist a year and a half ago. Now we have a cure, if you will, in the form of a vaccine or prevention, I guess you would say. But do you think we can take that lesson of developing a drug in 9 months or developing a vaccine in 9 months and apply it to other drugs and other therapies with critical unmet needs?
Yes, I think so. And for LMPs and vaccines, right? So big difference with developing biologics or small molecules, right? Where it's an enormous space. For nucleic acid technologies, the code is written. You just need to make it, right? So that cuts years of, drug design. You already know what the instructions are. In the case of, the spike protein for SARS-CoV-2, once that was known, you just make it, right? And so for diseases in which, you can apply these modular solutions, I think that you can move fast. And examples, some of my colleagues in the Oligotherapeutic Society meeting, we, as you know, there are multiple oligonucleotide type of drugs being developed, whether it's short interfering RNAs or siRNAs, or a single strand, antisense and splice switching type oligos. Those have been clinically validated. You can make it, the synthesis, the characterization, all of that exists. So people are now beginning to think about using that platform that exists for N of one type of, therapies, right? So for these hyper personalized therapies, we'll need to figure out as a field how to really execute through that, because it's today the cost and it's not there yet. But there is a really great example for that was the lead was Team U at Boston Children's Hospital, right? Where there was a patient with a rare disease. He identified the mutation and developed single-stranded oligonucleotide and started treating that patient. That was done. There is all of this is published, so I don't know exactly the time, but it was months, right, between the sequence and being able to treat that patient. So is it possible? Yes, it's possible. How much of that we're going to see being implemented, we'll need to wait and see.
Wow, my head's exploding with that. This is amazing. Really, really have enjoyed listening to all this science. I want to talk about mentors a little bit. That's something I try to hit on in all of my Molecular Moments episodes. And I have to believe that as a CSO, mentoring is a big part of that. And boy, I feel like you've taught me so much just sitting here today. So, and you mentioned it actually earlier, the mentoring that you received. Can you just talk a little bit about that in your experience there?
Yeah, so I think that mentoring is one of the great gifts that you can give someone and that you can receive, because here, people are sharing with you the wisdom of their knowledge and their experiences. And for me, it's one of the core components as a leader and as a people manager. So at Intellia, we are very keen in working with our teams, with mentoring, and also really elucidating different career paths, right? Because, sometimes, when you go to grad school or even in college, right, it's kind of, very limited what, the opportunities are, right? So for me, I had formal and informal mentors, throughout my career. I have a very close group of friends. We call each other the Wonder Women. We work together at Merck. We all come from different backgrounds and we help each other, and this is like a sounding board. One of the things I really enjoy with them is that, you can be completely open and vulnerable because that, they really want to be there for you and provide, this actionable feedback, right? So here is a problem. This is how I approach it. What did you do, not do? So, I encourage people to have this, group of people that are going to be with them throughout their career. So I think that's really, really important. The other is the misconception that in a mentoring, relationship, someone has to be much higher than, the mentor needs to be much higher than the mentee. And I don't think that applies, right? So I have gained a lot of knowledge from people I mentor, right? Perhaps more than what they gotten from me. So, and you could have, peer mentoring, which is, very important as well. The other is, I mentioned before, but is this concept of paying it forward. So as people have helped you, make sure you do that to others, and at the end, it all is a big circle, right? Because, it comes back to you at one point. Karma, good karma, bad karma too.
Just want to touch on one last point with you. Boy, we're going on about a year now of COVID and lockdown started in March. Anything you took on in 2020 or, over the course of COVID, anything you focused on? What's been your outlet here over the last year, almost year?
So the silver lining of COVID times was that I could spend more time with my family. And, I'm really grateful about that. Hobbies, I spend a lot of time in meetings, so I'm in my office. So, besides learning how to solve the Rubik Cube, which was something that was pending from my high school years, I guess, I've been doing, growing succulents, which is so fun. I mean, it's slow. I wish I could speed it up a bit, but, it's fun. And the other is using Zoom and Skype to connect with people. I've been a lot more intentional about, doing that. So, I've re-engaged with even many of my friends from high school. Now we have a debate club every month where we talk about different topics, and it's really fun.
That's fantastic. Laura, thank you so much for coming on and being a guest. I feel like I have a new mentor in the gene therapy space. I got so much out of this. Thank you so much. And I would love to have you back maybe in a year or two when we see where things have gone. We can get an update on Intellia's research. There's so much we could have talked for 2-3 hours, I think. I could have listened to you. So thank you so much for joining us today.
Yeah, and thank you again, Chad, for the opportunity. And I hope to see you in Boston sometime. And
absolutely.
Have a nice dinner.
Well, that's all for episode 6. If you enjoyed today's episode, be sure to subscribe on Apple Podcasts, Spotify, or your favorite podcast app so you never miss a conversation. If you'd like to hang out with us outside the podcast, we have many webinars and other presentations available for your enjoyment and education. Visit bioagilytix.com to see what's coming up and how you can stay in touch. And don't forget to keep an eye out for more episodes coming soon. We're looking forward to some great guests. We'll have world-renowned experts talking about rare diseases, diversity in the pharmaceutical industry, new and exciting technologies, and a conversation with a patient who has benefited from the recent tremendous developments in our industry. Molecular Moments would not be possible without the support of our sponsor, BioAgilytix Labs. BioAgilytix is a global contract research organization specializing in large molecule bioanalysis. Based in Durham, North Carolina, with labs in Hamburg, Germany, and Boston, Massachusetts, BioAgilytix provides high-quality bioanalytical services to leading pharma and biotech companies around the world. They offer assay development, validation, and sample analysis under non-GLP, GLP, and GCP, as well as GMP quality control testing. If you're looking to work with a team of highly experienced scientific and QA professionals through all phases of clinical development, look no further than BioAgilytix. For more information or to speak with their scientists today, visit their website at www.bioagilytix.com.
Thanks for listening to the Molecular Moments podcast.